Bismillahirrahmanirrahim. We come to the organization on and development of the nervous system. There are basically two major components of the nervous system: the central nervous system and the peripheral nervous system. The peripheral nervous system contains the nerve fibers contained in the cranial and the spinal nerves, and the neurons that make them up. And the central nervous system consists of basically all the neurons, their cell bodies that are derived from the neural tube. Now, all the cell bodies and the axons of the neurons that are present in the central nervous system, they are derivatives of the neural tube. But the peripheral nervous system consists of neurons whose cell body are present outside the central nervous system, and they are derived from the neural crest cells. So basically, in order to put it simply, we can say that the central nervous system consists of all the cells which are derived from the neural tube alone, and the peripheral nervous system, which consists of the fibers of the nerves. They are a mixture of the two. For example, you can see the skeletal motor neurons and the axons of preganglionic autonomic neurons. They are derived from the neural tube. And if you just try to concentrate on this point, is that the skeletal motor neurons, their cell bodies are present inside the spinal cord, which is a part of the central nervous system. But the cell bodies which are of these neurons are present inside the central nervous system. Their axons, they move out of the central nervous system into the spinal nerves, and they become part of the peripheral nervous system, and then they supply their target organ, which is the skeletal muscle. And the same is true for the preganglionic autonomic neurons. Their cell bodies are present inside the spinal cord, again in the intermediolateral horn of the spinal cord, and their axons, they move out of the central nervous system into a spinal nerve, and then they innervate the target organs. So actually the nerves contains the nerves, the peripheral nerves, they contain fibers which are a mixture of the two. Either they are derived from the neural tube, or they are derived from the neural crest cells. Another main difference is the type of glial cells. The glial cells present in the peripheral nervous system are the Schwann cells, and the glial cells present in the central nervous system are the oligodendrocytes. So the Schwann cells, they make myelin. Glial cells are the cells which make myelin, which myelinate the nerve fibers, the axons. They form a sheet of protein around the axon. So that is myelin. So the myelin formed for the peripheral nervous system axons is the Schwann cells. And the myelin formed for the CNS neurons, or the central nervous system neurons, is made by the glial cell, which is known as the oligodendrocyte. So the difference, again, one more difference is that the Schwann cells, they myelinate only a small part of a single axon, while the oligodendrocyte can myelinate many segments of an axon and many axons. So with these fundamental differences, we come to the development of the nervous system, and the development occurs with a process which is known as neurulation. Basically, neurulation means the formation of a hollow tube or a neural tube. So how that tube is formed through which the entire nervous system develops. This is basically the process in which a tube is formed, and that tube will give rise to the entire nervous system. So this process begins in the third week of development, And both the CNS, the central nervous system, as well as the peripheral nervous system, they are derived from the neural ectoderm. Remember this: all the neurons, all their cell bodies, all their axons, they are derived from the ectodermal layer, both the central nervous system and the peripheral nervous system. I can't stress it enough. But the notochord has to be present over there in order to induce the overlying ectoderm to form the neural plate or the neuroectoderm through if the notochord is a part of mesoderm, if it is not present, it will not be able to induce the ectoderm that is lying just above it in order to form a thickened plate of cells, which is known as the neural plate, and this is the neuroectoderm, and it will give rise to all the neurons of the CNS as well as the PNS. As it is shown in this diagram, we are looking at the embryo from above. It's cut open and we're looking at it from the above, and this is the rostral end, this is the caudal end, this is the notochordal process. This is the process that I was talking about. This is a part of the mesoderm that will induce the ectoderm which is present over it in order to become a neural plate. Now this is a thickened part, thickened layer of cells that's just above the notochord. See, this is the notochordal process, and it's part of the mesoderm. The red part is the mesoderm, the blue part is the ectoderm. As you can see, the blue part is the ectoderm, the red part is the mesoderm, and the yellow part is the endoderm. So the notochordal process is part of the mesoderm, and just the ectoderm just lying above it, it becomes a neural plate, a thickened layer of cells which becomes the neural plate. And the two ends of the neural plate, they become elevated. As you can see over here, these are known as the neural folds. They begin to elevate and they form the neural folds. And since these two ends they elevate, they form a depression in the midline. Because of the elevation of these neural folds, there's a depression in the middle line that is formed, and this is known as the neural groove. And the neural groove, it deepens as is shown in B, in the diagram labeled B, is that the neural folds, they deepen, they further elevate, neural groove deepens until they fuse in the midline. As you can see here, the neural folds have come together and they have fused in the midline, and the surface ectoderm has grown over it, and this is completely now surrounded by the mesoderm. Now this neural tube, it descends down. A neural tube is formed, a hollow tube is formed when the two folds meet together and they form a rounded hollow tube, and this is the neural ectoderm, or this is known as the neural tube, and it is inside the mesoderm. And there's also a portion of the neuroectoderm which forms the neural crest cell, as you can see over here. But as you can see, these cells are outside, they're lying outside the neural tube, so they will form a part of the peripheral nervous system, and all the central nervous system will be derived from the neural tube, and the peripheral nervous system will be formed mainly by the neural crest cell, as you can see. And this is the classical dorsolateral position of the neural crest cell, as you can see in this diagram in D, that they're present, their location, as you can see, they're present in the dorsolateral position surrounding the neural tube. Another point to note is at this early stage of development, you can see that the neural tube has a polarity already. The dorsal portion of the neural tube is known as the alar plate, and the ventral or the anterior portion right here is known as the basal plate. So all the primary sensory neurons, the neurons which are going to collect information from the outside environment, they will be restricted to the posterior portion or the dorsal portion, or the alar plate of the neural tube, and all the motor fibers, they will come out of the basal plate, which is the anterior portion. And these two plates are separated from each other by the sulcus limitans. And this plate basically is saying the same thing that I've already said, that the notochord induces the overlying ectoderm to form the neural plate or the neuroectoderm, and by week 3, neural folds grow over the midline and fuse to form the neural tube. This is the second step in the process, and uh, and during this closure, the neural crest cells also form from the neuroectoderm, and then the neural tube has polarity. As I said, the alar plate is the sensory plate, and the motor plate is the basal plate, the anterior part, and they are separated from each other by the sulcus limitans. And then the neural crest cells will contain, or they will give rise to all the primary sensory neurons and all the postganglionic autonomic neurons and other non-neuron cell types. The peripheral nervous system will consist of 12 pairs of cranial nerves and 31 pairs of spinal nerves. This is the general organization of the nervous system.